process to detect this analyte. And here we use the same device, the OFC paper device, to do some airborne exposure detection. We use a chamber. In this chamber, we introduce a lot of explosives in the air, and we try to detect based on like a small volume of the solution in the top of the electrochemical cell. You have good correlations between the experiments in solution and the experiments when we try to introduce more explosive inside the chamber based on the air approach. And other application, like a stripping analysis, we could use this type of approach to detect different types of ions in solution, or metallic ions in solution. And we could try to do this type of approach to detect based on the current of the device. And recently, other research that we are trying to do is try to combine this OFC paper with gold nanoparticles, and we are trying to use these gold nanoparticles to attach or try to immobilize these gold nanoparticles based on the fabrication of these gold nanoparticles in the not printed area of the OFC paper. Here is one of the devices, our first device. This is a very new result that I will try to show to you. We are trying to produce the device. Here's the position of the gold nanoparticles in the OFC paper. Here is the silver ink. In here is a piece of lead where you have a graphite, 0.5 graphite, on the top of this paper. We are trying to use this piece of paper is like only working as a electrochemical cell, like a reservoir. We are trying to use only here to put the solution on the top of this stripe, these 3 electrodes. Here are some results to compare or try to do a comparison between these 2 approaches. Here's a conventional gold electrode with the same geometric area of this other OFC paper with the nanoparticles, with the gold nanoparticles deposited in the paper. Here is to show that the current is very large between the properties of the gold nanoparticles. Here's an image of this type of structure. Here, in this approach, we try to use this electrochemical device using only a infrared light to dry the nanoparticles in the paper. But we are trying different types of approaches. One of our other approaches try to use ER, but we are trying to use temperature together with the ER light. And from these 2 approaches, it's possible to check that the current without the temperature is higher than the current that we obtain using the ER and the temperature together. We are trying to check what's the reasons to do that. We are trying to look for the images. Some structures are more like nanoparticles characteristic than the other ones, but we are trying to understand how this current could be enhanced based on changing of the properties of the materials. Using another background and try to use again paper, but now using colorimetric detection. One of our ideas here, based on these 2 approaches of the Susslich group in the University of Pittsburgh, we are trying to use, it was our first work introducing paper with colorimetric detection. In this paper, the Susslich group tried to use some dyes or metalloporphyrins, pH indicators, and like the base or acid pH indicators. And they tried to use this type of approach to use the change of colors here, to use like a molecular recognition. This change of color could show something like a fingerprint of the molecules or fingerprint of the solution. They printed these materials using a pin printing array, and with this approach, they introduced something in the literature, or they were the first researchers to report this type of approach. If we take a picture before the image and after the exposition of this type of material, we can do this mathematically, the subtraction of this image and this image to use this type of approach, like the difference map. The great idea, or one of the big ideas to introduce this remarkable approach is that images here, the subtraction of these 2 images, could work like a molecular fingerprint of the samples. Here there is, in this article here, and there are some different types of molecules, and these molecules have a single color or the single spot. Some spots could light it in different positions and in all of these images. This approach caught our attention because we are trying to reduce the cost of the device and try to produce a very cheap device. In this approach, they used organically modified silica, and one of our ideas why we could not use chromatographic paper to do the same or try to obtain the same results. And in this approach, in Susslick approach, they used a pin printed, and in our approach, our idea is try to use a box printing technology. And the Susslick approach, they use a scanner and ChemSense software. We need to pay for this software. But our idea is why not we use a smartphone technology with a homemade app to try to detect or try to develop some device that could be used by anyone that have a smartphone. Only briefly report, this survey, every 4 years we have a survey about what type of sensors the people would like to have in their smartphones. This is the last one, showing that the people would like to, for example, sensors for gases, sensors for proximity, which we have now, but they would like all other sensors are listed here, but they would like more sensors attached to the smartphones. For example, glucose, alcohol detection for the brief analysis, air quality. This is the other type of the people would like to have in their smartphones. This field is enhanced with the time. We have now glucose sensors. We have pH sensor attached to the smartphones for the Android phones, for the iOS smartphones. And one of our ideas, why we not use the paper device together with the smartphone camera to try to detect explosives. This is one of our ideas here. We try to use a paper printed with wax. We immobilize some three different reagents here, and we took pictures before and after from this piece of paper. And we developed an app here to try to use to try to create a graph that's very easy to detect. Not very easy mathematically, but we try to use principal component analysis to do this type of approach. Here's how we produce the device. We use the paper, chromatographic paper here. We try to use wax, temperature to produce the spots, and we mobilize these three different reagents because each one of these reagents could change the color for one type of explosive that we are interested in. We are interested in three different types of nitro explosives and two different types of peroxide explosives, THP and HMTD. And why we use these reagents? For the peroxide explosives, because of the composition to produce peroxide, in this presence of these compounds, we could produce a brown coloration. And Frenchman tests, we do a little bit different approach with this type of test, but we try to use these compounds need to be used in the Frenchman test in acetic acid, but we try to use only in acidic medium to try to produce, or this mixture to produce within the presence of nitrobenzene and aniline, a yellow color. And we use Jaffe reaction, very useful for detect creatinine, but we are trying to use creatinine to detect prickly acid. The idea is try to took some images before and after the exposition. We produce for this type of device to maintain the optical distance the same. We use this device attached to this chamber. This chamber is to control the focal distance and the light that I will show in another slide. And here is the software that we use. We changed it a little bit, the software with the years, but the idea of here is try to extract some information about the colors, RGB colors, and we have the standard deviation and the mean value for this type, for all of these three parameters. With these parameters, we introduce these parameters in this matrix, and this matrix is analyzed by the PCA approach to obtain these different types of clusters, showing that the samples analyzed are different from each other. Here's some images from the, the chamber. We have a chamber here produced by a laser cutter. And we have here this chamber with these 4 LEDs positioned here to control the light. Here are some images, or RGB images, from before and after images and different map produced here. And it's possible to hear, not very good because of the light here, but all of these images are a little bit different that produce a unique fingerprint from the samples. Here in the real images, it's better to check this difference. Here are before images and after images. In the presence of these 5 explosives, we have some different images produced here. And with this cheap and simple device, we could produce, we can discriminate between these 5 explosives, and we can characterize why these clusters are positioned in different types of regions of this graph. And with the dendrogram, you can check that all of the explosives could be separated between each other. We have a good discrimination of the samples. It's not a real approach, but it was requested by the reviewers of the articles. And if someone tried to mixture different types of explosives, it's possible to detect this. This is the answer for one of the reviewers, that we can produce different clusters, or we can check different clusters, different positions, showing that it's possible to discriminate between these different types of approaches, or these different type of mixtures of explosives. Here's our same quantitative performance. I will not talk a lot of this equation, but we can use a mixture of a univariated approach with a multivariated approach. Here, these equations are not like the univariated approach. It only works in these regions. And these regressions do not cross in the 0, point. This is normal for this type of approach. And here is some quantitative parameters. We could detect very low amount of explosive using a single piece of paper and a smartphone detection. One of other ideas is try to use this type of approach. It's another new approach. We are trying to use, not now, for the paper, it works for using paper device too, but one of our ideas here is try to produce these materials in the same material used to cover the meat. And we try to use the smartphone technology to try to detect bacteria in meat using this type of approach. Here's very briefly, we changed a little bit from cellulose to cellulose acetate. It's a large difference, but the idea here is try to use this type of material because of the food compatibility, we produce these acetates, this is cellulose acetate materials using 2in20, and we immobilize some dyes in these membranes. The idea is try to detect amines because all of these microorganisms release these amines. And here's the production of the device. We have some plastic membranes with different types of dyes immobilized. And we took images before and after. And with these images, we try to discriminate between as the same approach that we use for the paper device. Here are some real images. We have a large difference between these amines detected here. Here, these 3 amines could be discriminated here with the same approach, with these 5 dyes used here. Here's is a single test only to show that it's possible to regenerate the material. If this material was left, it was left without the amines, it's possible to reuse this material. Here's a real sample with a real test with meat samples with these different type of amines, but with these images here. But one of the tests that we are very, very excited to do is, well, and if you use this to detect real bacteria, and we try to use the same device to detect these 4 bacteria here, all of these bacteria are pathogenic bacteria that you would like to try to understand if it's possible or not to detect this type of bacteria. And here are some real images, before and after, and difference map for these bacteria. It's possible to check that all images are a little bit different from each other for if you use just different types of bacteria. Here's the PCA plot showing that for very similar optical images, which is impossible to detect what type the bacteria is, but the electronic device or the colorimetric device could be used to detect all of these bacteria based on the amines release. Here's a same test using not now 37 degrees, but using 25 degrees, a real experiment. This is a KNN classification. I will skip from this to try to show other things interesting. In one of these approach in our lab, we are trying to use a forensic device to detect cutting agents in cocaine. Is one of our ideas, is a joke here, but one of our ideas not to produce a very good cocaine sample, but our idea here is try to track the origin of this cocaine based on the use of these cutting agents. And to try to do this, we have a lot of different devices. I'll try to show this a little bit faster to show all of the devices. We use some background from the literature, but we use this paper device to detect, based on the change of color, 4 types of cutting agents. One of them is phenacetin, that's a little bit one of the large used by the counterfeits in Brazil. And with this approach, we have a good detection limits, and we have a not large amount of interfering species. We have, for the phenacetin, we have some problems with the paracetamol, but we are trying to circumvent this problem of the interference. Another approach using cobalt reaction, using for the levamisole, we are trying to have good results, very good linear results, to detect from a large range of concentration. And in other approach here, we have no interference for the levamisole test. And one of the other approaches that can be faster, because Thalita will show these results for you tomorrow. And here we have an approach that we try to combine colorimetrics and electrochemistry. We use a reaction to detect here procaine. Procaine gives us this color here. But one of the main problems that we have with cocaine, we have good linear limits, we have small limits of detection, but we have a very, very big problem with the benzocaine. And one of our ideas try to how we can remove this interference. We are trying to use electrochemical sample pretreatment, and we remove one of the interference, and we can remove the benzocaine interference here. Thalita will talk a little bit more about this with showing videos, but this is one of the devices that we attached the pretreatment samples with the colorimetric analysis to produce a single device with all of this type of measures, and Thalita will talk a little bit more about this. Because one of our ideas here is we try to use this device now for a colorimetric approach to use by the forensic people or forensic researchers. They could apply here the cocaine samples, and we can get a fast answer about what type of cutting agents will be used, or it was used in this type of approach or in this type of analysis. And one of other approaches when we try to combine the electrochemical measurements and colorimetric ones, one of our ideas, or for a future, is try to attach or try to put different types of sensors in different types of river, and to have a very fast answer about the water quality. The water quality, the parameters used here in Brazil, it's very difficult to obtain. We need to do a lot of different types of analysis to obtain these numbers here, and these numbers here could be used to show to you if the water quality is good or not. Our idea is try to do the same. We would like to extract information for the river. We didn't know how much we have of some analytes in this river, but our idea is try to use the PCA to show if the water is good or not based on this type of the measurements. This is our idea that I was talking about. We have some previous results showing that if you use microfilms, golden microfilms, or gold electrodes, we can extract electrochemical information. But the mainly point here is if with this briefly, or with this very first results, we can classify different types of water very fast without to quantify any of these analytes here. And one of these ideas is try to put this in a single device, but electrochemical could be good until a single point. But our idea is why not we can use the combination of electrochemical cells with the colorimetric analysis. We can do a stripping analysis here, and we can use these zones here, these detectant zones here, to detect the samples that are not be, or that could not be detected by an electrochemical technique. This is an approach for the iron, and you can analyze the other metals here. Here's some electrochemical curves here, and some relative intensity curves here for the colorimetric device. The same approach could be used here for metal detection and nitride detection. We could apply here to detect the metals, and we could use the detection zones to detect nitride here. We can do this for multivariate analysis. Probably you think that I like to use multivariate analysis sometimes. And one of these ideas is try to use single colors to measure pH with a good fit for this type of approach for this pH that's mainly used for the environmental analysis. And as conclusion, one of the ideas here that I try to show to you is the paper device has an experience helped go over the past decades. And paper device could be used not only for the point of care device, but other areas. And try to show to you that the combination of different types of techniques could be useful to try to solve some approach on some real approaches. And I would like to acknowledge my students. Without them, it's not possible to show off these results here. And I would like to show off these guys here, because without money, we don't have anything to do in the lab. Thank you for the attention.
Thank you, Thiago. We have time for 3-4 questions. No questions?
Thank you very much for a very impressive presentation. Very nice work. There's been a lot of work in the paper-based diagnostics field. And I always have the question, who's going to make money here? Because it's zero cost approximately. So is there any business that you can take out of this?
Mainly we don't have any necessity to have money to do this. But we have money to support the students, support the infrastructure of the lab. This money is necessary sometimes for this type of approach. And sometimes we are trying to combine this type of techniques with high cost techniques, because there is some approach to try to combine paper device to introduce samples in the mass detection. Sometimes we need some money for this type of approach. But if you think of these small projects, don't give for us a large money, but a small amount of money that could be used to produce this type of articles.
Is there an option to look at food safety, if it's still okay to eat it or drink it? You can print on the etiquette something like this. Is this some business model? Or?
This is one of the ideas. There is a water forum that we are trying to do some work together with the French group. We are trying to use this type of device to be attached to the water bottles, different types of bottles to detect very fast something about the quality of the water or something else.
Okay, thank you.
Any question?
Nice talk. I was wondering about, imagine you have tried to develop some electronic nose, because I'm looking that, I think the dog it's very hard to get the same sensitivity with a dog nose. But imagine if you have some devices that you can replace, in some cases, to find explosives by volatile compounds. You have some idea, or you think about this?
Yes, yes. We have some people in my lab working with this type of approach. Because I started with the nose, with the tongues, because I have more experience with measures in liquids than in air samples. But we are trying to work to try to detect different type of cancers based on the breathed air. Because it's easy to detect acetone or other compounds in the breathed air, and try to use this type of devices or this type of molecular fingerprint made on paper to try to detect different types of cancer based on the breath. We are trying to do this based on the idea of the dogs and on the smell, something.
So no questions? Last one.
Thank you, Thiago. Very nice talk.
Thank you, Murilo.
Glad to see such nice application, different applications. And paper start with different fabrication techniques. It has evolved to different types of application, integration. And such a more a general question, in your opinion, what's gonna be the next step on paper-based devices?